animal house designing, breeding and maintenance of animals, production of transgenic animals. Dear students, in this class, let us learn about animal house designing. Introduction. Animal facilities built to appropriate architectural and engineering standards are expensive to build. The finished structure should reflect the future and present needs of research. The collective outcome must be an enhanced research environment and the uninterrupted facilitation of excellent animal care practices. It requires integration of information which will drive the programming and designing and construction. Animal house is a place where certain laboratory animals like rats, rabbits, guinea pigs are kept in controlled condition for study, research and experimentation. Proper housing and management of animal facilities are essential to animal well-being. The environment in which the animals are kept should be appropriate to natural conditions. A well-planned, designed, constructed, properly maintained facility is an important element of good animal case. The design and size of an animal house depends on the scope of institutional research activity. Effective planning should include input from personal experience with animal facility design and operation from representative users of the animal facility. Good animal management and human comfort and health protection require separation of animal facilities from personal areas. For an ideal animal house design, one has to follow CPCSEA, that is the Committee for the Purpose of Control and Supervision of Experiments on Animals, rules of Department of Environment, Government of India. Functional imperatives of animal house facility. The purpose of a laboratory animal facility is to confine animals in comfortable, safe, stable environment that are conducive for research, teaching and testing. These animals often vary in their microbial background and facilities should therefore incorporate barriers and routines to minimize cross-contamination. Location. Laboratory animal facilities should be located to facilitate the receipt of animals, supply after breeding as well as the removal of waste, and should be located to preclude both public access and need for movement of animals and dirty both through public areas. Animal houses must have access to reliable services including water, electricity and sewage disposal. Animal houses must be located so as to ensure access to a high quality source of air. Materials and finishings. Materials and finishes should be durable, resistant to water and chemicals used in their sanitation. In addition, they must be resistant to damage by equipment used in the facility such as cage racks. Crevices, cracks and unsealed service penetration that can harbor diet and vermin should be eliminated, or all hollow doors must be filled or completely sealed. Walls. Walls should be covered with an impervious coating that withstands frequent cleaning and chemical disinfectants. The walls should be free of cracks. An epoxy coating is frequently applied. Floors. The base floors for animal house should be concrete slab. Seamless epoxy flooring with integral cove base is the most common flooring used, especially in animal rooms. It is durable, impervious to many chemicals and solvent-based products, and easily cleaned. It can be made less slippery by adding grit to the surface. Ceilings. The preferred substrate for ceilings is moisture-resistant drywall that is well sealed at all ceiling wall joints and penetrations. Doors. Top quality doors and frames made of durable metal should be used to prevent access to vermin. Windows in the doors are extremely useful to allow observation into rooms and as a safety feature. The direction in which the door swings should be such that they are safe, do not impede traffic flow and complement the control of airflow. Interlocking doors are often useful to ensure that only one door is opened at a time. Self-closing doors work effectively. Windows. Natural light is beneficial to animals. Windows are not recommended in the most cases for animal holdings rooms due to the difficulty they pose in controlling internal environment. Windows are incorporated into outside corridors. Plumbing. Potable water must be available within the animal facility. Ample hot and cold water must be available throughout the house for sanitation purposes. Water must be available for all safety equipment, emergency fire sprinkler systems. Sink showers must be present to minimize the potential for contamination. Automatic flushing systems should be installed. Electrical outlets. All lights fixtures through the animal room should be vapor-proof. Cage washers, autoclaves must be installed according to recommendations. The electrical and wiring requirements for security monitors must be considered. Temperature, relative humidity and differential pressures should be monitored frequently in each and every animal holding room. All safety equipments must be installed. Equipment that generates large amounts of noise should be isolated from the animal house. Animals sensitive to noise should be located far away from noise-generating equipment. In most animal rooms, lighting should be designed to provide at least 2 levels of intensity during the light cycle. Diurnal light cycles in animal houses should be controlled and monitored centrally. Heating and air conditioning. It should provide healthy, comfortable environment, clean air, specific temperature, humidity continuously. Maintenance of animals. Introduction. It is important when designing animal rooms to consider not only current needs but also possible future requirements. In most animal facilities, animal use fluctuates according to changes in research projects. A versatile holdings room will facilitate rearrangement in order to accommodate the cage racks and equipments to house different species. Versatile grouping of rooms will permit a variety of projects to be undertaken over a number of years. The ability to modify environmental parameters is also often necessary to accommodate various research needs. Animal holdings rooms. Separate animal holding rooms should be available for each species, each group of animals of different health status within a species and different animal use where the care and use regimes differ significantly. The size of the animal holdings room should be determined by the species, the number of animals to be housed, the type of housing, the proposed animal use and the services needed. The room should hold the animals comfortably in a suitable environment with sufficient space to service animals. Ventilated cage racks that supply ventilation individually to each cage on a rack are being used more frequently to house rodents. Mobile hoods are used. Procedure rooms. Invasive procedures that may cause distress to other animals should be conducted in a procedure room. Those include injection, collection of blood samples. Well-established procedure rooms should be available within the animal house to reduce the need to transport animals to laboratories located outside. Surgery. Surgery must be performed under aseptic conditions, like surgeon preparation and scrub animal preparation and pre-medication, surgical operation including anesthesia and post-operation recovery, intensive care, surgical storage, equipment storage and servicing, surgical preparation and sterilization. Clean and dirty loading docks. The receipts and disposal of clean materials and dirty materials should be segregated. The loading dock must be designed to restrict the entry of vermin into the animal facility. Animal reception areas should be separately ventilated. Feeding and bedding storage. Animal bedding must be stored in a vermin-proof room and should not be stored directly on the floor, but rather on plastic or metal pellets or shelves. Waste storage. The waste storage area must be large enough to accommodate all waste accumulated between disposals. The ventilation system for the waste storage area must be designed so that exhaust from this area cannot enter any part of the building or adjoining buildings. Biohazardous waste, hazardous materials and waste containing radioactive substances must be stored separately. All waste products must be eliminated in a safe manner. Equipment must be incorporated, ensure safe elimination of waste. The disposal of cage and dirty activities in the cage wash area must be segregated. The differential pressure on the dirty side of the cage wash area must be strongly negative to all surrounding area. Mechanical cage washers must be used. Clean equipment must not be stored in corridors or animal rooms. Sterilization. Equipment should be installed within the area in which it is used at transition zones. Physical and chemical sterilization are practiced. Necropsy. Incorporation of a necropsy area is recommended for health surveillance system. A detailed post-mortem examination is often required for scientific purpose. Corridors must be wide enough for sufficient protection, and other critical areas such as surgical and necropsy suites, current biosafety guidelines must be consulted in all cases where animals are infected with known human or animal pathogens. Breeding of animals. History of animal breeding. The different breeds and strains of domestic animals have developed over thousands of years during the process of evolution in wild and domesticated stage through intensive system of production to meet the marketing demands. They must have gone through stress conditions for developing different genes and gene combinations in them regarding their adaptability to different geoclimatic conditions, resistance to diseases, and heat tolerance. The natural selection and genetic drift caused the breed differences. Man's attempt to influence the evolutionary process of domestic animals by selecting the animals with desirable characteristics. Migrating and crossing to produce desired animals over a period of time also caused breed difference. All these caused the evolution of new types of animals with distinct appearance within a species. Pure breeding in humans is an ancient idea for a number of reasons, particularly the caste systems in society. Man's attempt to breed livestock to serve his purpose goes far back into man's own history. The use of pedigrees was started in rural England late in the 18th century. Robert Bakewell, 1726 to 1795, is remembered for setting the pattern of modern animal breeding and is called the founder of animal breeding. He made efforts to produce farm animals like cattle, sheep, and horses with increased efficiency. Inbreeding produces prepotency and refinement and development of relatively true breeding strains. Types of breeding: inbreeding and outbreeding. Inbreeding. Inbreeding is a mating system when the mates are more closely related than the average members of the breed. The mated individuals should have one or more common ancestor in their pedigree up to 4 to 6 generations. This is because the contributions of ancestral genes to the total genetic constitution of the descendant of the 7th generation is less than 1% on the basis of halving nature of inheritance. Types of inbreeding. Inbreeding upon the extent of relationship of the mated animals can be said as close breeding and line breeding. Close breeding is the mating of more closely related individuals like parent-offspring mating. Close breeding is done for some specific purposes like to develop highly inbreed lines, to discover undesirable recessive genes with the help of parent-offspring mating, to get more uniform progeny. Close inbreeding has certain disadvantages like inbreeding depression and intensification of undesirable characteristics. Line breeding is taken in two ways. The first way is mating of animals which are more distantly related like cousins mating. In this system, the rate of inbreeding is less than close breeding and hence the inbreeding depression is less. The second way is the mating of animals in such a way that their descendants are mated to outstanding animal up to 3 to 4 generations. The line breeding causes a rapid increase in homozygosity. The aim of line breeding is to perpetuate the special character of an outstanding animal or strain. The prime aim of line breeding is to maintain a high relationship to an outstanding ancestor for retaining its good proportion of genes. Genetic effects of inbreeding. It increases homozygosity at the expense of heterozygosity without affecting the gene frequency. Inbreeding increases the frequency of recessive traits in the population. Inbreeding fixes character in an inbred population due to gene fixation, which may be favorable or unfavorable. The homozygous animals, particularly for dominant genes, are more prepotent than non-inbreds. Inbreeding reduces genetic differentiation between lines. Phenotypic efforts of inbreeding. Inbreeding has an adverse effect on fitness traits like reproductive efficiency in terms of delay in puberty and testicular development, reduction in gametogenesis, and increase in embryonic losses. Inbreeding results in increased death rate and loss of vigor, vitality. Inbreeding also causes certain recessive genetic defects and some abnormalities are expressed. Applications of inbreeding. Inbreeding is a useful tool in animal breeding which eliminates the undesirable genes. Inbreeding is used to produce distinct families within a breed. Inbreeding is used to develop inbred lines as a seed stock and helps to know the type of gene action affecting a trait. Outbreeding. The mating of unrelated individuals is called outbreeding or genetic disassociative mating. The unrelated individuals may not necessarily belong to the same herd or different herds of the same breed, that is outcrossing. The offspring formed by mating to unrelated parents is called hybrids and the phenomenon is called hybridization. Intraspecific outbreeding. It is the mating between the members of the same species. Interspecific outbreeding. It is the mating between the members of different species. Intergenic outbreeding. It is the mating between the members of different genera. Crossbreeding. The mating of animals from different established breeds is called crossbreeding. Crossbreeding is practiced for commercial production of livestock. The two breeds differ genetically and hence the crossbreds are expected to show hybrid vigor or heterosis. This makes the crossbred more productive and better than the parental breeds. Crossbreeding combined of livestock can be evolved. Crossbreeding is also used to study the behavior of character in hereditary transmission. Types of crossbreeding. There are two basic methods of regular crossing known as specific crossing and the rotational crossing. In specific crossing, two pure breeds may be crossed or may be extended to crossing the crossbreds, intersmating, or with males of pure breed, that is back crossing. In rotational crossing, males of 2 or 3 breeds are used in regular sequence in successive generations. Genetic and phenotypic consequences of outbreeding. Outbreeding increases heterozygosity. Outbreeding increases the height, viability, fertility, resistance to environmental factors, diseases and pests. Recognized animal house and animal breeding are suitably approved by Animal Ethics Committee of the department consisting of members with main and link nominees selected by CPCSEA New Delhi. Production of transgenic animals. Genetically manipulated animals having an introduced gene are called transgenic animals or transgenics. Transgenic mice, sheep, cattle, goats, pigs, poultry and fishes have been developed by using genetic engineering techniques. The introduced gene expresses its character in the transgenic and is inherited for many generations. Transgenic animals produce some novel proteins of therapeutic value, more milk and show fast growth. 1. Transgenic mice. The genetically engineered mouse is the transgenic mouse. The desired foreign gene can be transferred to mice by retroviral infection, microinjection and transfection. In the retroviral method, the desired gene is inserted into a retroviral DNA and the retrovirus with recombinant RNA is made. The recombinant virus is allowed to infect 8-celled embryo and the embryo is implanted in the uterus of the female mouse. The mouse produces transgenic siblings. In microinjection, the foreign gene is directly injected into male pronucleus in the fertilized egg. Then the zygote is implanted in a female mouse. In transfection method, embryonic stem cells, that are the ES cells, are taken from a blastocyst and the foreign gene is introduced into the cells by transfection. The transfected ES cells are introduced into blastocysts taken from a female and the blastocysts are implanted in the female. Uses: Ralph Brinster in 1982 developed transgenic mouse by introducing rat growth hormone gene. The transgenic mouse siblings attain 44 grams weight in 10 weeks, while the normal siblings are 29 grams. They are called super mouse. Transgenic mice are used as human disease models for AIDS, diabetes, cancer, and immune functions. They are also used to study gene regulation, immunological reactions, and tumor development. They are used to assess the feasibility of drugs to use for human diseases. They are also used to produce proteins of therapeutic value such as CD4 protein, urokinase, etc. CD4 protein used for AIDS treatment. Urokinase is used to dissolve blood clot. 2. Transgenic cattle. Mature oocytes are collected from cows are fertilized in vitro by semen collected from a bull. The fertilized oocytes are centrifuged to settle the yolk at one pole of the oocytes. Then the foreign gene is micro-injected into male pronucleus in the oocytes. The oocytes are grown in vitro till they attain blastocytes stage, and then the blastocytes are implanted in the uterus of cows. The cows produce transgenic calves. Many transgenic cattle strains been developed by introducing different novel genes. 1. Transgenic cows with K-casein gene insert produce milk rich in K-casein. This milk is likely for cheese making. 2. Transgenic cattle with lactase gene insert produce milk which is poor in lactose. This milk is good for lactose intolerant people. 3. Transgenic sheep. Mammary glands of the transgenic sheep serve as bioreactors and produce some proteins of high therapeutic value. Example: alpha-1 antitrypsin, that is ATT, factors 9, etc. The alpha-1 antitrypsin gene is fused with a beta-lactoglobin promoter and is micro-injected into male pronucleus of fertilized egg. The egg is then implanted in the uterus of a surrogate mother. The sheep produces transgenic lambs. The transgenic lambs, after reaching maturity, give 1 to 35 grams of alpha-1 antitrypsin in a liter of milk. Emphysema, a lung disease in man, can be treated by injecting alpha-1 antitrypsin. Transgenic sheep with gene for factor 9 produces factor 9 in the milk. Factor 9 is isolated from the milk, purified and used to treat hemophilia in man. For having high rate of wool production, sheep have been genetically manipulated to have growth hormone gene or keratin gene or cysteine gene. The transgenic sheep produce more high quality wool. 4. Transgenic goat. The transgenic goat with TPA gene produce tissue plasminogen activator in the milk. The TPA is used to dissolve blood clots. 5. Transgenic pigs. Gene for factor 8 is introduced into zygote of pig by micro-injection and the zygote was implanted in the uterus of a sow. The sow produced transgenic pigs that could produce factor 8 in the milk. The factor 8 has been isolated and purified from the milk and used to treat hemophilia. The transgenic pigs with human beta-globulin gene produce human hemoglobin in their blood. Attempts have been taken to use the hemoglobin for transfusion. A team of researchers at Immutran Company USA micro-injected certain genes from human immune system into male pronucleus of pig zygote. They introduced the zygote into a sow to produce a transgenic pig. The transgenic pig had human proteins so that human body may accept their organs. The transgenic pig was named Astrid. Attempts have been taken to transplant heart, kidney, liver, lungs and pancreas of transgenic pigs in human beings. 6. Transgenic rabbit. Transgenic rabbits have been created by introducing interleukin-2 gene along with a beta-casein promoter. They produce interleukin-2 in the milk. It is used to treat cancers. Transgenic rabbits having human growth hormone gene grow faster and produce more meat within a short duration. 7. Transgenic birds. Some blastocytes are isolated from freshly laid egg and are transfected with desired gene through liposuction. The transfected blastocytes are then introduced into blastoceles of irradiated eggs. The eggs hatch into chimeric chickens. If the blastocytes had entered the germline, the chimeric chickens breed transgenic chickens. Transgenic chickens resistant to avian leukosis virus (ALV) and certain bacteria are developed in this way. Efforts have been taken to increase the meat quality of chicken and to reduce fat and cholesterol in eggs. 8. Transgenic fishes. Desired foreign gene is introduced into 4-celled fish embryos by microinjection. The embryos are grown into transgenic fishes in temperature-controlled tanks. Transgenic fishes such as carps, catfishes, salmons and tilapia have been developed by using this method. Transgenic goldfish was made by introducing human growth hormone gene into immature embryos. They grew faster than the normal goldfishes. A gene for antifreeze protein (AFP) of a winter flounder was transferred to Atlantic salmon. The transgenic salmons can tolerate low temperatures. 9. Transgenic mosquitoes. Gene coding for the endotoxin of Plasmodium vivax was introduced into zygote of mosquitoes by microinjection or transfection. Transgenic mosquitoes developed from the zygote produced toxin of malarial parasite in their saliva. When the transgenic mosquitoes suck blood, they inject the toxin into the human blood. The human immune system produces antibodies against the toxin so that the body resists the malarial parasite. The transgenic mosquitoes are used to develop transgenic mosquitoes to vaccinate people against many bacterial and viral diseases. Conclusion. The animal house is carefully designed for the maintenance of different animal groups approved by CPCSEA, New Delhi. Utmost care is taken to maintain animals in animal house, which is meant for study, research and experimentation. Breeding of animals brings about high growth rate, hybrid vigor, heterozygosity and homozygosity in animal population. Transgenic animals are organisms containing integrated sequence of cloned DNA transferred by using the techniques of genetic engineering. These animals have not been released easily as a release of conventional animal breeds. They are restricted strains which have been handled with care and released after so many objections. Thank you.